Video of the day. So, muscular system review. All right, so we got a lot that we're going to go through. This will be the longest one, I think, of all the review ones. If you are struggling with just the slides that I put and you want a little more detail, let me know and I can direct you back to the PE 220 videos. All right, so we've got muscle anatomy. All right, so when we get down to it, the functional unit of a muscle cell is called a sarcomere. All right. Now, within this sarcomere, we have several different proteins. So the first one is myosin, which is this purple stuff. Where did my highlighter go? I've lost it. There we go. So purple right here. Ooh, that green doesn't work real well. Okay. And what it does is that it moves the actin. So actin are these little, the green circles here that you can kind of see. And then we've got troponin, which moves tropomyosin out of the way, ultimately allows actin and myosin to bind. And then we've got titin, which is an elastic protein that we'll learn about more during the neuromuscular unit. Its job, at least as far as we've discussed so far, is that it helps stabilize actin and myosin. Okay, and then we've got, and it helps get the muscle back to resting length. It's basically this springy thing on the end of the myosin. Okay, and then we've got nebulin, and nebulin is an inelastic protein that also helps with structure, and it stabilizes actin. And so within a sarcomere, we've got a couple of different zones or bands, and so we've got the I band, the A band, and the H zone. Okay, now if you look, the A band is wherever there is actin, the I band is wherever there is the A band is wherever there is myosin. The I band is wherever there is no myosin. And then we've got the H zone, which is only where we have myosin. Now, if you notice when we go from up here, we've got a relaxed muscle, to down here where we've got a contracted muscle, notice that the A band stays constant because the length of all of these proteins doesn't change. The only thing that's changing is that we're pulling actin towards the middle, and so the overlap changes. And so in this case, the I band gets shortened, and that's this part here, okay, and the H band shortens, okay, and this is because of the overlap is changing between actin and myosin. All right, and so we've got a neuromuscular junction, and so this is where a neuron talks to a muscle cell or is able to communicate with a muscle cell. And so you can see up at the top here, we've got this brown thing is our neuron, and then the red part is our muscle. So we have an action potential comes down, releases acetylcholine. That acetylcholine binds to receptors that ultimately cause an action potential to occur. Okay. And within that, ultimately what we're doing is we're releasing calcium so that it can bind to troponin, so that tropomyosin moves out of the way so actin and myosin can get together. We call this process excitation-contraction coupling. So here's just another picture that shows it in a different way. And here are the steps. Now, I'm not going to go through each of these steps, but it is expected that you understand and know how this works. Okay, if you have questions or you're unsure or you don't remember or something doesn't make sense, please ask that question in class. Now with excitation-contraction coupling, we've just got actin and myosin bound together. Okay, so we've got what's called a cross bridge. And cross bridges are important because the more cross bridges we have, the more force that we can generate. But we're not shortening the muscle here. So basically myosin has to pull actin to the middle, release, grab onto another part of actin, pull it to the middle, release, so on and so forth. It's kind of like a tug of war. And so this is where the sliding filament theory occurs. Okay, and we'll hopefully demonstrate this in class to some extent, but really it is like a tug of war. And so what's happening is you have something going on with ATP, which is going to cause a change in myosin and actin. And really it's causing a change in myosin that will potentially affect actin. So when ATP binds to myosin, actin and myosin separate. When ATP breaks down, the energy causes that myosin head to cock. And in doing so, actin and myosin are actually reattached. The phosphate leaves, and this causes the myosin head to go through its power stroke. And so this is where we're pulling actin towards the middle. The ADP leaves, actin and myosin are tightly bound. Okay, that is the sliding filament theory. Again, this is review. If you need more information or whatever, please let me know. But that's trying to keep it as simple as possible. Okay, so then we've got fiber types. And we've got 3 different fiber types that we're going to talk about, but please know there are many more than 3. But basically, we're going to go from type 1, type 2A, and type 2B. Type 1 is essentially aerobic, needs lots of oxygen, so needs things that will help it use oxygen. So lots of myoglobin, lots of mitochondria, lots of capillaries, but it's slow. It doesn't produce a lot of power, but it has a lot of endurance. Whereas type 2B or type 2X are the exact opposite. It fatigues really fast, it produces a lot of speed and power, but it gets tired quickly. It doesn't use oxygen. And then we've got type 2A, which is kind of a combination of type 1 and type 2B. Now, in order for a muscle to contract, it has to be receiving a signal from a neuron. And so a motor unit is a motor neuron and all the fibers it innervates. Now notice we've got a muscle here, and within that muscle are many, many fibers, okay? And we've got one neuron that's innervating 3 of these fibers and one neuron that's innervating 2 of those fibers. However, we've got 3, 6, 8, 9, so we're accounting for 5 of these muscle fibers, but not all 9, okay? And so when a muscle contraction occurs, we might not actually be activating all of the muscle fibers. As a matter of fact, we are never activating all of the muscle fibers. All right, and so it's important to remember an individual muscle is made up of multiple motor units, and you're going to have different types of motor units, so different type 1, type 2A, type 2X. Now, with motor units, the way they work is through the all or none principle. If a motor unit is excited, it's going to do its job, and it's going to do its job to the max. All innervated muscle fibers will fire maximally. Okay, if nothing happens, if threshold doesn't happen, nothing happens. Okay, so it's kind of like a gun. Either a gun fires or it does not. Like you can't half shoot a gun. Okay, so again, we come back to this picture. We've got a couple of motor units, but we've got many, many, many muscle fibers. And so when you're just doing something that low intensity, okay, so when I'm doing, like I'm curling, like a clicker in my hand, my whole muscle is getting shortened, but I'm only activating a few of the motor units and a few of the muscle fibers. All right. When I'm like maxing out on a bicep curl, I'm activating nearly all of my motor units. And this relates to the size principle. We recruit motor units in the order of their size. So we go the smallest to the largest. And the reason that we do this is so we don't use our large fibers when we need small force. The example I use in PE 220 is I like to make cookies. I usually do this about once a week. My wife and I do this. We keep it on the down low. And so if I was grabbing an egg with my strongest motor units or my biggest motor units, I'm going to break that egg. That's not going to be great. So this allows us to have fine motor control. So the last thing that we've got here is, or not the last thing, the next thing is the different types of contractions. So we've got isometric, which is constant length, so like pushing against a wall, or the muscle contraction that's going on while you're sitting there, and the isotonic, which is the tension or the force you're producing is constant, but the length of the muscle is changing. Now with isotonic contractions, they can either cause the muscle to shorten, which we call a concentric contraction, or we can call this an eccentric contraction if the muscle is lengthening. Okay, so think about it with a bicep curl. When you do that curl, when you're having elbow flexion, that's a concentric contraction in your bicep. However, when you bring the bar back down and you increase that joint angle, in other words, you're having elbow extension, that's an eccentric contraction of your bicep. Now, when it comes to the length tension relationship, or in other words, how long is the muscle, how much force can it produce, it is dependent on the length. And if you notice, the greatest force that we can produce is that kind of resting length. But as we increase the length, we produce less force. As we decrease the length, we produce less force. And the reason being has to do with cross bridges. As I increase the length, there are more there are fewer cross bridges, okay? You've got like at the longest length of the muscle, you only have a couple of cross bridges that can generate force, and so I'm unable to produce as much force. Whereas if I've decreased the length of the muscle, yeah, I've got lots of cross bridges, but I've overlapped so much that I can't actually pull that muscle, make that muscle any shorter. The last slide is talking about how much force we can produce and how fast can that muscle contract. Now, when we're doing a maximal contraction, the velocity is 0. Okay, when we're doing essentially no contraction or have no force, we can go really, really fast. So the way I want you to think about this is if you've ever done a bench press and you're maxing out, how fast is that bar moving? And the answer is it's not moving very fast at all. Okay, eventually you're going to get to a point where the bar just kind of freezes and it's sitting there. And unless someone is spotting you and picks that bar up, it's going to go back down on your chest, and that's less than ideal. So the faster you have a muscle contraction, the lower the force that you can produce. And that's where we're going to end for muscular system review.